Thank you, David, for inviting me and organizing this really exciting meeting here. And I will give you a few ideas and thoughts about our view. What is the problem in biology? So we are thinking that the biology is somehow generally upside down. And the problem is that our sciences started with a very complex system, as we humans are. But normally in science, you should start with simple systems. And when you understand simple systems, you should go up and up and up, higher. And the problem now is that all these really like basic terms like memory, learning, cognition, sentience, and so on are mostly reserved for humans, and it makes a lot of trouble somehow to use these terms in other systems. Although they are in history, there were many people, important people, who were somehow convinced that the life generally is based on cognition and sentience. And recently, Arthur Reber, and I joined him later, we started this new concept, which is called Cellular Basis of Consciousness, or CBC in short. And in this concept, in fact, we claim that cells are the primary organisms which should be first understood and studied. And this is in fact, a bottom-up approach to consciousness. And in our view, the plasma membrane, which is excitable and makes the border, intelligent border between the inside and outside, in some term it is in fact the Markov blanket, is the critical structure for generating and using sentience in cells. We also think that the circadian clocks, which are cognitive and anticipatory systems which evolved with the very first cells, and these cells were some kind of sentient cells, some 4 billion years ago. And these are based on a redox system, the most ancient part of these clocks. Of course, later the gene expression patterns were also integrated, but on the basis is the redox code, which is the basis of these circadian clocks of cells. And we think that these clocks, based on redox code, are closely linked to the cell sentience. In fact, in a eukaryotic cell, you have several cells, in fact, because eukaryotic cells is composed at least of 2 cells, but we think it is more. And in plant cells, we have plastids, and every organelle even is having its own clock. So in fact, the eukaryotic cell clock, circadian clock, is composed of several subcellular clocks, and the organismal clocks then somehow is integrated from all our cellular clocks. How? It is really a mystery how this happens, how this could happen. And so we think our ideas are somehow linked to the Barbara McClintock, who was, I think, one of the very first one with very important statement that every cell is having a knowledge about itself, and every cell is using this knowledge in a thoughtful manner when challenged. But then after Barbara McClintock was Lynn Margulis, who published a paper 2001, where he proposed that the bacterial cell is having conscious faculties, and also she proposed that due to this symbiotic origin of the eukaryotic cell, the bacterial consciousness is very complex. And we have also some other important experts here. Pamela is in fact sitting here with us, who were some of the first who proposed that the prokaryotic cells, bacteria, are having cognitive faculties, and the life and cognition is absolutely inherently linked together. So in our concept of the CBC, we believe, or we propose, that the outside-inside dichotomy is the most important issue, and it generates the inside as a subjective field, and outside is the objective field. And we propose the concept of synome, which is based on the plasma membrane, and it generates movable and ordered charge molecules, and also different radical molecules which are moving around. And we propose that this is somehow creating something we call synome. And the synome, in our concept, is a model of the cell, which cell has inside of the outside world. So the survival is possible only if the synomic information is really having high fidelity. So in some way, those cells which were having not high fidelity were somehow weeded up, and the membrane synome is mostly linked to the sensory even and the plasma membrane, and the synome is also inherently linked with the cytoskeleton in eukaryotic cell, of course, but also in bacterial cells. So we propose this view of a cell as a triadic nature of a synome in blue. Sorry. So, wait. Oh. Sorry. Yes, with the synome in blue, the epigenome in yellow, and the genome in red. And we have, for example, cells which have very high synomic activities, like, of course, neuronal cells, which is based on the rapid recycling of endocytic vesicles, which increase the space or the border between outside and inside. And for example, red blood cells is having very low activity, so it would be on the opposite pole with very low synome, and in fact, red blood cells doesn't need too much information about the environment for its purpose. Oops. It works now. Can I move it here? Okay. So this slide is, I think, the most important from all of I will show today. And in this slide, the most important issue is that the life on the Earth started some 4 billion years ago, and cells are ancestral cognitive organisms. And due to the fact what was first defined by Virchow, but shown by Remarque and Rispile, is that there is no de novo formation of new cells. So every cell must come from the previous cells. So we think in a time, some 4 billion years, how many cell divisions were there. So it is almost not possible to count. And there is some kind of a history in the cell structures. So the structure of the cell is inherited, and so is also the order inherited via these cell divisions. So it means we can also say, like Virchow was saying, omnis cellula cellula, omnis synome synome, because you cannot generate synome de novo. It can come only from previous synome, and we think it is a very complex order inside of this synome. And cells have, due to the synome, a sense of a self, and they can act as a cognitive systems due to this synome. So I would like now to also mention that sometimes the evolutionary theory is having troubles because it seems that the cognition is not taken into account. And Karl Popper was one of the first who in this lecture, Organisms Generate Knowledge to Solve Problems, somehow was tackling this problem. And also recent paper by Eliezer et al., they showed, which was nicely commented by this commentary article, that the organization of the neurons and the neuronal circuits is modified by sensory experiences. So there is a feedback from our perception, or in this case it was a C. elegans, that we can modify our brain, our networks, neural networks, just by our sensory perceptions. And now I'm going to go a little bit to the plants, because in plants we have something similar. Here you have two plants, Arabidopsis thaliana, having the same age, same genome, growing in the same space with same watering. The only difference was that this plant here, the smaller one, was touched once a day. Just brief touch, and that's all. And after a few weeks, you can see a huge difference between these two plants. So this sensory perception of the touch completely changed the phenotype of this plant. Again, evidence that the sensory perceptions can control phenotypes also in plants. We published this paper in 2009, which was one of the out-of-the-box paper, and it created a lot of criticism, where we showed using this MEA apparatus, which is multitude electron array system, that in maize root apex we have a zone which is having very high activities of spikes. This zone is called transition zone, and this is one of the examples that the cells together can coordinate their electric activities and to produce some some extracellular field of electric field, which I show you soon. And also it is one of the examples, I would say, of the in-space epicenter in the root apex. In root apex, in this transition zone, we showed that it is very sensitive to anesthetics. So if we apply anesthetics on plants, plants are really affected strongly. Endocytic vesicle recycling is inhibited, actin cytoskeleton is disintegrating, and the plant cells cannot generate spikes, action potentials. And in this zone, we have very specific arrangement of actin, which is shown here, which we published some 25 years ago, which can be found only in these cells. Here is shown schematically. And in this zone, the electric fields around the root apex is also having inward pattern in contrast to other zones. So root apeces are generating electric fields which can be measured with electrodes, similarly like it is in brain when we can measure our activities with EEG system. So it seems that all cells together can generate a coherent, much stronger fields which can be measured, even with our techniques which are still not very sensitive. It will be better maybe in 10 years. So this is again an example of N-space epicenter of the root apex. And so actin cytoskeleton is very often forgotten in respect of action potentials. But what is important is that it is not only cytoskeleton for moving organelles, but it is also having polyelectrolytes features, which is meaning that along actin filaments, ionic waves are running and even electric signals. So in fact, this element of cytoskeleton acts also as some kind of transmission structures. And all cells are sensitive to anesthetics, which is somehow related to the actin cytoskeleton also. And in this transition zone, which I was mentioning already, we have a synchronous oscillations of anatomically grouped cells, and this is very similar to the definition from neuronal system where synchronous oscillatory patterning of anatomically grouped neurons are driving sensory motor networks in brains. So in roots, we have something very similar. And again, if we apply anesthetics, organ movements are also stopped like it is in animals or humans. And also in plants, I don't have time to go into this in detail. We have all the neuronal molecules like glutamate, GABA, we're having very similar functions as in animals. One issue here with the cancer cells is interesting, that cancer cells are known to have very low membrane potential, the lowest, in fact, from all cells. And maybe this is the reason why they are out of the control of the body and they can act as a very bad cells for our organisms. Interestingly, the plant cells are even having high membrane potentials like neurons. So we have scores up to 200 millivolts in plants, which is much more like in neurons even. So now I would like to go to anesthetics, the last part of my talk. Claude Bernard was somehow very important in this respect, and he was making a definition of life on the basis of anesthetics. What is living can be anesthetized, what not is dead. So according to his definition of life, everything can be anesthetized, and this was really shown later. It was supported by many, many papers. He was doing experiments even with the mimosa plant, as you can see here. And he was also using animals, and he showed that same concentration of anesthetics are causing anesthesia in animals and plants. We have published recently one paper, but now there are several already, where we showed that we can anesthetize different kinds of plants, like mimosa, dionea, or drosera. We can also use local anesthesia with lidocaine applied on the roots, and this will anesthetize also shoot part of the plant. And what is also interesting, the anesthetics are having also anti-cancer and antimicrobial effects, and this is logical because all cells are sensitive on anesthetics, so even the bacteria or cancer cells are sensitive. And the next very important issue for me is that they also affect circadian clocks of cells. So again, this indicates that the circadian clocks based on redox code and xenom based on also redox code are somehow closely linked together. So this is a summary where we show that every cell, even the symbiotic mitochondria or plastids have their own clocks. They are integrated somehow into one unicellular clock, and all these unicellular clocks are then integrated in humans or in plants into the organismal clock. How this is done is very difficult to explain, but somehow it seems to be related to the fact that also every cell is having own sentience, and this cellular sentience is somehow merging together and generating the organismal sentience of our bodies. Of course, we are not aware of, about our cell's sentience, because it would be completely making our life not possible if we would be aware of all this. So in evolution, our brain is for us the most important for our consciousness. And I would also advertise the book which will be published soon. Arthur Miller is also here in the audience, where we put together all these ideas, and especially we are explaining how cell sentience is generated and how cell sentience is important for understanding life. Thank you very much.
Okay, we have time for questions. Any questions? Okay, do we have, oh, yes.
Yeah, thank you for your talk. So what about populations of cells that are microorganisms? How far can you push your ideas?
Yes, definitely there are trillions of microbes in our gut and stomach. They have definitely their own consciousness, sentience, but again, as I tell you, even every our cell is having its own version of sentience, but our brain is not getting it directly. Of course, you can have a gut feelings, you can have subconsciousness, there is something, but it is not easy to, understand.
So I have a question. So I understand the experiments of Bernard and you that you can anesthetize the plants and other cells, but how does that susceptibility, so it indicates that cells have electrical potentials?
No, the evidence that cells have electrical potentials and action potential was done many times and not related to anesthesia. It is well-known fact.
Right.
It was published several times. What is quite strange, in 70s, the plant electrophysiology was getting really troubles because it was somehow considered to be exotic science. And there were many groups of plant electrophysiologists, but they were all died out. And I think one of the last one is a group of Rainer Hedrick in Würzburg. He also published now several papers on anesthesia. And yes, it is a big problem now because this field is completely wiped out.
Yeah. And so my question is, how does the effect of anesthesia on these cells imply sentience? I didn't follow that.
If we have anesthetics and we are lying somewhere in the hospital, so we cannot react actively to the environment. And the same is with the plants. So they are in some kind of a sleep status. They are not able, of course, the metabolism of cell is running, but if you would increase concentration, this would be also stopped. And that's why we have anesthetic experts who must dose you perfectly according to your age and weight.
Yeah.
In order that just your brain is out, but that your body is still functioning. But of course, if you increase the concentration, you would wipe up everything.
Sure. Right. I see. Okay. Are there a remote? Emily.
Thank you, Frantisek. I was fascinated with the section on the genome and its genesis in redux, taming and dealing with these poisons that are generated in the course of metabolism. Has that influenced your thinking about what sorts of information processing hardware might have evolved first, like ion channels first, or signaling
Yes,
proteins first, or that sort of thing?
I think first in the protocells or coacervates, first I think there was some protocinome there, even without any membrane, but this protocinome was not, couldn't reproduce because without membrane and cell division, it was just coming, going, and it took some time until the first plasma membrane, which was able to persist, and the cell division was somehow invented, that the genome and all cellular structures started to be inherited in the cell division. And of course, I think the genome came, I think, later. So the genome is all the time upstream. So if you have some sensory impact on cell, genome reacts in milliseconds. Epigenome maybe a few seconds, and in genome there will be some changes in minutes. So the genome is the upstream. I think it's the primary system of the cell.